RNA-Seq library prep turns RNA into sequenceable DNA. First, extract RNA. Then enrich mRNA and fragment it. Convert it to cDNA, add adapters, amplify, and sequence. Millions of reads later, you get gene expression data. First, cells are lysed to release RNA. This includes mRNA, rRNA, tRNA, everything. Next, RNA is separated from proteins and DNA. Then, it is purified to remove contaminants. Finally, the RNA quality is checked to ensure it's ready for sequencing. But not all RNA is useful. We usually focus on mRNA, the part that codes for proteins. So we either pull out mRNA using poly-A tails, or remove abundant rRNA. In this step, long RNA molecules are broken into smaller pieces. This is important because sequencing machines can only read short fragments, not full-length RNA. Fragmentation can be done using heat, magnesium ions, or ultrasound, which physically or chemically break the RNA. As a result, we get many short RNA fragments. These fragments are the ideal size for sequencing and will be used in the next steps to build the RNA-Seq library. RNA is fragile, so we convert it into cDNA using reverse transcriptase. A primer binds to the RNA, and reverse transcriptase builds a DNA strand from it. This creates an RNA-DNA hybrid, which is then turned into double-stranded cDNA. cDNA is more stable and ready for sequencing. Special adapter sequences are added to both ends of cDNA. These adapters allow sequencing, sample identification, known as barcodes. The library is amplified using PCR to generate enough material for sequencing. Finally, the library is loaded into a sequencer. Each fragment is read, producing millions of short sequences. Millions of reads later, you get gene expression data.